Hi everyone, welcome back to Dr. Han's Pharmacy Classroom Pro. Now in this video, we are going to have a quick review of the basic anatomy of skeletal muscle structure at the tissue level. Then I'm going to go over the actin-myosin action behind skeletal muscle contraction. At the end, let's review the events that happen at the neuromuscular junction. So let's get started. Now for those of you who are not a vegetarian or a vegan, the meat of what we eat comes from animals' skeletal muscle. Now, it doesn't matter if it is a chicken leg, beef sirloin, or pork chop. They are all just a selected cut of skeletal muscle from the respective animal. Now, for those of you who also like to cook, you may have noticed a thin layer of wrapping surrounding the meat when you are trying to cut it. Now, that piece of thin wrap is called the actin-myosin. Now, the actin-myosin is the outermost layer of connective tissue that surrounds the entire skeletal muscle. It is a dense layer of collagen fibers that provides structural support and protection to the muscle. The actin-myosin connects the muscle to the surrounding tissues and helps transmit forces generated by the muscle contractions. Now, under the actin-myosin is the perimyosin, which is a connective tissue layer that surrounds bundles of muscle fibers called the fascicles within the skeletal muscle. And fascicles are groups of individual muscle fibers that work together to generate force. The perimyosin contains collagen fibers, blood vessels, and nerves that supply the fascicles. It provides structural organization and support to the muscle by compartmentalizing the muscle fibers into functional units. The endomysium is the innermost layer of connective tissue that surrounds individual muscle fibers or muscle cells within a fascicle. Now, it is the most delicate layer and is composed of fine collagen and reticular fibers. The endomysium supports and separates individual muscle fibers. It contains capillaries and nerve endings that supply nutrients and signals to each individual muscle fiber, allowing for communication and control of muscle contractions at a very fine level of detail. Now again, for those of you who have eaten chicken legs before, don't we all hate the chewy part that connects the leg meat to the bone? Now that's the tendon. A tendon is a tough and fibrous connective tissue that attaches muscle to bone, and it is composed of mainly of collagen fiber. And tendons are designed to withstand tension and force, allowing muscle to pull on bones and create movements. Now, tendons are designed to withstand these forces, allowing it to create some great movements. So if it breaks, that would be pretty terrible. Now let's enlarge the scale even more and look at an individual muscle fiber. Again, the endomysium contains capillaries and nerve endings that supply nutrients and signals to each individual muscle fiber. And within the endomysium, there is a layer called the sarcolemma. The sarcolemma is the specialized cell membrane that surrounds individual muscle fibers, which are the individual muscle cells that make up skeletal muscles. And a myofibril is a cylindrical thread-like structure found within muscle fibers that makes up the contractile machinery of skeletal muscles. Now, I know my illustrations of the muscle cell now looks like a pepperoni pizza. Now, from this angle, the yellow cheese-looking area would be our T-tubules. A T-tubule is also known as transverse tubule. Now, it plays a crucial role in transmitting electrical signals deep into the interior of the muscle fiber. T-tubules are invaginations or inward extensions of the cell membrane or the sarcolemma that extend deep into the muscle fiber. They run perpendicularly to the longitudinal orientation of the muscle fibrils and allow for rapid transmission of action potentials from the sarcolemma to the interior of the muscle cell. Another structure at this level is the sarcoplasmic reticulum, or the SR. Now, one of the primary functions of SR is to store calcium ions. And when a muscle is at rest, the sarcoplasmic reticulum actively pumps calcium ions from the cytoplasm, or the interior of the muscle cell, into its lumen. During muscle contraction, an action potential travels along the T-tubules and reaches the sarcoplasmic reticulum. Now, this triggers the release of stored calcium ions from the SR lumens into the cytoplasm. The presence of calcium ions in the cytoplasm initiates the interaction between actin and myosin filaments, leading to synchronized myofibril contraction. The terminal cisternae are expanded regions of the sarcoplasmic reticulum that are located adjacent to the T-tubules in the skeletal muscle fibers. The arrangement of a T-tubule sandwiched between two terminal cisternae is often referred to as a triad structure. Now let's look at the mechanism of muscle contraction, or the actin-myosin action. Within the myofibril, there are several key components and structures. First, we are looking at the sarcomere. The sarcomeres are the repeating segments within myofibrils that give skeletal muscles their stripped appearance under microscope. Now, sarcomeres are bound by Z-lines. These structures serve as anchor points for the thin filaments of actin. Actin is a protein that forms thin filaments within the sarcomere. These filaments are anchored at the Z-line and extend toward the center of the sarcomere. And actin filaments are essential for muscle contraction as they interact with myosin filaments to generate force. And myosin is a motor protein that forms thick filaments within the sarcomere. And myosin molecules have protruding heads that interact with actin filaments during muscle contraction. And M lines are found at the center of each sarcomere, serving as a reference point for the arrangement of myosin filaments. And during contraction, actins move toward the M line through a sliding movement. The H zone, or H band, is a region within the sarcomere where only myosin filaments are present. During muscle contraction, the H band narrows as the actin filaments slide inward, overlapping with the myosin filaments. The I band is the region between the two adjacent sarcomeres, contains only actin filaments. It becomes narrower during muscle contraction as the sarcomeres shorten. The A band is the central region of the sarcomere that contains both actin and myosin filaments. It remains constant in width during muscle contraction. And the Z-line comes closer together as sarcomere shortens. This shortening of sarcomeres contributes to the overall shortening of the muscle fiber, leading to muscle contraction. Now let's look at the actin-myosin action at the protein level. The muscle contraction requires energy in the forms of ATP, and ATP is associated with the myosin before contraction. ATP is just like money. Money only has value when it is spent, and ATP only has value when it is split into ADP and inorganic phosphate. Now this activates the myosin into a standing position. It binds to the binding site on actin and slides the actin toward the M line, producing the contraction. Now let's chronologically look at the actin-myosin action. In a resting muscle, the actin and myosin filament are positioned so that they partially overlap, but there is no direct interaction between them. Calcium ions are stored in the sarcoplasmic reticulum and the muscle is relaxed. And when a motor neuron sends an action potential to the muscle fiber, it travels along the sarcolemma and T-tubules. This triggered the release of calcium ions from the terminal cisternae of the sarcoplasmic reticulum. And calcium ions bind to troponin molecules on the actin filaments, causing a conformational change in topomyosin, which is a protein that covers the active site on actin. The conformational change in topomyosin allows the active sites on actin to be exposed. These active sites are binding sites for myosin heads. And myosin heads, which contain ATPase enzyme, binds to the exposed active site on actin, and forming a cross bridge between actin and myosin. Now, when ATP molecules bound to myosin are hydrolyzed, releasing energy and causing the myosin head to change shape, this change of shape is referred to as the power stroke. During the power stroke, the myosin head pulled the actin filaments toward the center of the sarcomere, causing the sarcomere to shorten. And after the power stroke, ADP and inorganic phosphate molecules are released from the myosin head, but the myosin head remains attached to the actin filament. A new ATP molecule binds to each myosin head, can cause the myosin head to detach from the actin filament. The hydrolysis of ATP into ADP and inorganic phosphate provides the energy necessary for the myosin head to return to its original resting position. During this process, the myosin head becomes re-energized and ready to form another cross-bridge with actin if calcium ions and active sites are still present. Now, as long as calcium ions are present and ATP is available, the cycle of cross-bridge formation, power stroke, and detachment repeats. And here are the short key points of the actin-myosin action. First, by calcium release, calcium binding to troponin, and myosin head binds to actin. Myosin move towards the M line, and then the ATP binds to myosin again. Myosin detach from the actin, and the cycle repeats when the ATP being split into ADP and inorganic phosphate again. Now let's look at the neuromuscular junction, which is the final part of this video. The neuromuscular junction, or NMJ, is a specialized synapse or connection point between a motor neuron and a skeletal muscle fiber. The motor neuron originating from the central nervous system, sending nerve impulse or action potentials down its axon, and the terminal end of the axon forms numerous finger-like structures called axon terminals. The synaptic cleft is a tiny gap that separates the motor neuron terminal from the muscle fiber's sarcolemma or cell membrane. Now, it contains gel-like substances that help transmit chemical signals. And the region of the muscle fiber's sarcolemma that is opposite the motor neuron terminal and is called the motor end plate. Now, it contains receptors for a neurotransmitter called acetylcholine. When an action potential reaches the motor neuron terminal, calcium influx to the terminal triggers the release of acetylcholine into the synaptic cleft from the synaptic vesicle within the terminal. Acetylcholine diffuses across the synaptic cleft and binds to the acetylcholine receptors on the motor end plate of the muscle fibrous sarcolemma. The binding of acetylcholine to its receptor on the motor end plate causes the opening of sodium channels and influx of sodium. This leads to a change in the sarcolemma's electrical potential, resulting in the generation of a muscle action potential. Now, this action potential triggers the release of calcium ions from the sarcoplasmic reticulum, initiating the sliding filament mechanisms and muscle contraction. After acetylcholine has transmitted the signal across the neuromuscular junction, an enzyme called acetylcholine esterase breaks down acetylcholine in the synaptic cleft, preventing prolonged muscle contraction. In summary, in this video, we have looked at the basic anatomy of skeletal muscle structure at the tissue level, the actin-myosin action behind skeletal muscle contraction, and the events that happen at the neuromuscular junction. That is everything for this video. Let me know if you have any questions, and I'll see you next time. Take good care. Bye.